David. So we switched to English.
So thank you, Oliver, for the invitation, and obviously congratulations for the designation of an excellent center. So I feel particularly honored to really participate in this process, because the fact that I have taken advantage sounds a little bit weird outside of the English, but taken advantage of really the excellent work that is conducted here at the University of Cologne many years ago when we worked together, and I'm still doing it, so we're still collaborating with you. And congratulations really to ECMM for this great idea of the network of excellent centers. So I think that it's a great initiative there. So what I'm going to try to share with you over the next few slides is really the highlights of a new product that we are developing with Scynexis. The name of the product is SCY-078. We call it 078 as a short way. And just to give you a sense, Scynexis is an organization in the US. It's a small biotech organization. We have about 16 employees only, so it's really a small organization there. Also, it has a long history, really focusing on the development of this particular compound, starting in 2014. And the compound was actually discovered at Scynexis in collaboration with the work that was conducted with Merck. And it was discovered as part of a platform or fungal derivatives that have a glucan synthase inhibitor. So the intention was we want to have a glucan synthase inhibitor that its structure allows for oral administration. That was the main principle of how this originated. We are a public company, and we're based in Jersey City. This is a little bit the highlights of the product, and as you can see here, the molecular structure is basically, it's a molecular structure that you can see here that is very much different to the molecular structure of the echinocandins. That will allow you to understand why the properties are so different. Even if we engage in the same enzyme, so we target the same enzyme for inhibition of glucan synthesis, we engage the enzyme in a different way. And that allows to have a slightly different profile in terms of resistance. And that also allows the product itself to have good oral bioavailability. So we are certainly a glucan synthase inhibitor with an IC50 of inhibition very close to the IC50 for the glucan enzyme, very close to the candins. And however, we are less impacted by the most common FKS mutation. And when I'm talking about B, it's basically the product. And we have good oral bioavailability. Key attributes of the product: it has a broad spectrum of anti-candida activity. Basically, all the strains that are covered by the candids are covered by 078 and probably a little bit more. It has a spectrum of activity against aspergillus as well, and it does have activity against pneumocystis. And so it's active against the majority of the Echinocandin-Resistant strains and certainly against azole-resistant strains. We are developing oral intravenous formulations, and so far we have exposed a little bit more than 300 patients and subjects, and conducted several phase 1 studies, including drug-drug interaction studies, and we have been able to confirm that the potential for drug-drug interactions is very low. Another interesting feature of the product itself is really the extensive volume of distribution. So once the product gets into the plasma, it distributes very efficiently into the tissues. And in many instances, achieving concentrations that are substantially higher than the concentrations in plasma in key tissues like lung, liver, spleen, vaginal tissue, mucosa. The product does not penetrate into central nervous system. So that's probably a limitation there. Considering the attributes of the product, to give you a picture, where do we think that this particular product could provide a benefit on? What are the type of indications that we're focusing on? Certainly we consider that a product with these characteristics could provide a benefit in invasive candidiasis, because it has activity against resistant strains. So that's something that we always want to have, something that is with the potential to address that particular need. And then it will allow a glucan synthase inhibitor that has demonstrated to be so far the most important or the most effective mechanism in treating invasive candidiasis. It will allow an easy transition from IV drug to the oral drug without really sacrificing any efficacy. And we consider that the product could also provide some utility in aspergillosis. And in this particular case, probably an alternative approach to improve outcomes, particularly in invasive aspergillosis, in combination therapy with azoles. We don't believe that a glucan synthase inhibitor by itself will ever be or is likely to be better product by itself versus an azole medication in invasive aspergillosis. Is unlikely that that will happen, and that's not going to be our intent or initial focus. So we're figuring out what is going to be our focus in invasive aspergillosis, but certainly combination therapy sounds attractive here. In chronic, though, it's an interesting alternative for azole-resistant strains, because since we have oral formulation, it can be given for long periods of time. In vulvovaginal candidiasis, we know that there are several limitations. There are not too many products approved for the VVC, and we certainly consider that this particular product having cidal activity against Candida has an opportunity in treating VVC and recurrent VVC. And also in prophylaxis. Having a spectrum of activity against Candida, Aspergillus, Pneumocystis with low risk for drug-drug interactions. We consider that this could be a good alternative in some of your prophylaxis needs in these regards. Very quickly, I'm just going to provide a highlight of what has been the recent data that has been published about our compound, what we have been frequently or recently done. So this is just to exemplify how the 078 behaves somehow different than the standard echinocandins or the candids, particularly against those strains that have FKS mutations. I don't know how familiar you are, but the FKS mutations are the main cause of echinocandin-resistance. Once the Candida develops some of those very specific mutations, often they do develop resistance. And as you can see here in this graph, that was a recent publication by Dr. Fowler. You can observe here that 078, the bar that is in the dark gray, are those strains that behave as non-wild type. So it behave as a resistant strains. And all these strains were tested for FKS mutation. However, up to 70% of the strains actually continue behaving without problems. They continue behaving with the good MICs in the same way that the wild type population is. But if you look at the different candids, they obviously, the presence of the FKS mutation substantially affects them more. So increases their MICs substantially more than it does for 078. So this is an interesting feature. Another interesting recent publications about our product is that certainly the product affects Candida auris, has activity against Candida auris. And images are very fine in this regards, but obviously you can see here that the image labeled as A is Candida auris in normal with no exposure to drug, and then you see the image in B is Candida auris with exposure to 078 at a concentration of 0.5 milligrams per liter. So you can really see that the Candida there doesn't look very happy. And also what we did is in a collection of 100 isolates of Candida auris, you can see here that the MICs of 078 showed evidence of activity against this isolate. And interestingly, in that particular table, you can see that even those strains that were tested and confirmed to be resistant to echinocandin, they maintain, I would say, a very favorable MICs for 078 as well. Another interesting feature is the fact that 078 has activity against biofilm. This is in an in vitro testing. Dr. Jesus Guinea, that is also a collaborator with ECMM as well, he tested for us the activity of 078 against biofilm. And as you can see here in these images, you can see Candida albicans, a very nice and healthy biofilm on the photographs labeled at A, whereas in C, you can see that really most of the biofilm has been destroyed here on activity of 078. That is an important feature that obviously we know that many of the Candida infections, invasive candidiasis, either are caused or are perpetuated by the presence of biofilms, and it is particularly important in certain conditions. So we have characterized the in vitro activity of 078 in several tests, as you can see here. As well, we have characterized the in vivo activity of 078 in animal models. The first way that you figure out if this particular product is going to work after you see it in vitro, then you move it to in vivo. And then the most common animal model for invasive candidiasis is the mouse model. And we have been able to characterize the activity of O78 in several mouse models and to actually identify what is the intended exposure in order to achieve efficacy. With this intended exposure, then you went to humans and you figure out, can I achieve that exposure in humans to be able to achieve that efficacy? And we're happy to report that we have been able to achieve those target exposures in humans with the oral formulation as well as the IV formulation. So what do we know about Aspergillus activity? Well, the O78 has an antifungal activity, in vitro antifungal activity against Aspergillus. I would say that very comparable to the other candidates with similar MICs in this regard for in vitro. It does have activity as expected against azole-resistant strains as well. And we conducted this experiment with Dr. Mahmoud Ghannoum, recently published or presented at ECMM, in which you can see here we were trying to verify if O78, how it will perform in vitro in the presence or in combination with isavuconazole, voriconazole, or amphotericin B. And as you can see here, and I'm not going to go to all the details, but you really measure how the product behaves by itself, then in combination, and then you determine if this is a synergistic activity or not. And you can see here that for the 3 drugs, in the great majority of the strains tested, the product showed a synergistic activity in vitro. So that sounds to us like an interesting development. Then, as I mentioned to you, you go in vitro and then you go in vivo. And this is a neutropenic mice model of disseminated aspergillosis. And in this mice model, we were able to also identify that O78 this is the control and this is survival. So the majority of the control animals died as suspected. And certainly the 2 doses that we tested here with oral O78, they showed substantial improvement in survival compared to the controls. And in this particular, to the untreated controls, and comparable to caspofungin and ambison in this model, that they both work very well. So what we did later, after that, we demonstrated that the product seems to have good activity by itself. How about in combination then? And this is a recent report from Dr. Tom Walsh in Cornell University, in which you can see here that we used the neutropenic rabbit model, and we gave the product in combination with isavuconazole. We use a dose of isavuconazole, which is typically compared to the human dose, to the human levels that are achieved with the standard dose. And you can see here that with isavuconazole, certainly, well, all the animals without treatment died. And with isavuconazole, the animals had very good response here, 33% survival, which is in line to what has been reported with this specific model. But with any of the combinations, we observe up to 66% survival. So this data is very intriguing to us, and we as a company are in the process of really assessing what are we going to be doing with this potential interesting effect in Aspergillus as well. We have conducted a series of drug-drug interaction studies, and also we have evaluated the QTC potential for the drug. We know that the only currently available oral antifungal medications belong to the azole family, and some of their limitations are drug-drug interactions and QTC propagation. So any new potential drug in the armamentarium should ideally kind of overcome these particular problems with the azoles. So O78 doesn't have any clinically meaningful effect on CYP substrates. The one that in vitro showed to be the worst was 2C8, and we actually conducted a clinical trial identifying what was the maximum effect in a substrate of 2C8, and we saw no changes, no clinical significant changes in the pharmacokinetics of the drug rosiglitazone. We also conducted a study, a drug-drug interaction study with tacrolimus, and the level of modification of the tacrolimus values were substantially low, 0.4, which was the best azole in the market, which is isavuconazole, potentially in terms of interaction, has an interaction of about 2-fold. We didn't see, O78 is not significantly affected by CYP inhibitors, and we also didn't have any clinical meaningful effect in a very extensive series of phase 1 investigations. No evidence of QTC effect here. Oh, that code messed up. Sorry for that. Here, putting into this format consolidated messed up. But anyway, we conducted a phase 2 study in invasive candidiasis. The purpose of this phase 2 study was a PK study. The main question was that we wanted to know if these doses that in healthy volunteers have shown to achieve the target exposure, we're going to be able to achieve the target exposure as well in patients with invasive candidiasis. And the answer was yes. So we were able to achieve the target exposure with a 750 milligram once a day dose. At the same time, even if this wasn't a small study, we wanted to have a check about the efficacy. We didn't want to have obviously a substantial different effect than the current standard of care. In this study, just to remind you, the patients were receiving echinocandin therapy. In average, they received about 5 days, and then they were stepped down to an oral therapy, and they were randomized in this fashion, as you can see there. Either 2 of the doses of oral 078 or fluconazole. And the efficacy that we observed, evaluated as a global response at end of therapy, I would say that they are comparable. This is a very small study as well, but what we were very happy to see is that we didn't see any. We didn't see any breakthrough infections while on therapy. So that was very, very encouraging. We do have an ongoing clinical trial in the United States, and we call it FURY. And actually, we are very pleased and thankful for the collaboration with the clinical trial at the Cologne Center, which is helping with setting up this trial in Europe. And this is a phase 3 study. It's an open-label study in patients that are refractory or intolerant to another approved drugs. And it's currently with the oral formulation, and we may include the IV formulation in the future. We are wrapping up our phase 1 activities with the IV formulation. And for instance, just to give you an examples of patients that could be included here. So patients with invasive candidiasis, including CRs, that have clinical or microbiological failures under standard of care. Patients for which oral step-down therapy from an IV echinocandin is desirable. However, their isolate will not allow them to safely be switched to an oral azole because of the high MSIs to fluconazole or risk of DDIs. Extended therapy is needed, and with a glucan synthase inhibitor, or severe mucocutaneous candidiasis like an esophageal candidiasis that is not tolerated. Anyway, we wanted to set up this trial in order to provide access to 078 for the treatment of these difficult to manage fungal infections, and at the same time evaluate the safety and the clinical utility of 078 as a salvage therapy in those very severe conditions. We are also doing another clinical trial. This is right now being run only in the United States. It's a phase 2 study, dose-finding study in patients with vulvovaginal candidiasis. And we are planning to have next year a global study, which is going to be against first-line therapy for invasive candidiasis. We are just expecting to complete the phase 1 for the IV development, so that both the product is going to have IV followed by oral, compared to IV echinocandin followed by an azole as a standard of care. And I think that as a summary, what is 078 is a novel oral IV glucan synthase, oral and IV glucan synthase inhibitor. The spectrum of activity is a broad anti-candida activity, good anti-aspergillus activity, and anti-pneumocystis activity. Extensive tissue distribution, and target exposure attainable with well-tolerated doses with low risk for drug-drug interactions and QTC effect. And we're very, very excited about the opportunity to really continue moving this product along and to bring it into your future armamentarium of drugs. Thank you.
Thank you very much. Thank you. So that really is a promising compound, and it's a mode of action where we are familiar with. So yeah, it'll be good to finally no longer be forced into changing the drug class just because you go oral and things. Thank you very much, David.